आती है।
अस्सलाम वालेकुम सर।
बेटा today we will start new chapter. It is cell membrane and transport across cell membrane. Okay, so just give me a minute, let me get ready. Okay, in this chapter we will discuss fluid mosaic model. Okay. And then we'll discuss the components of a membrane that include cholesterol, glycolipids, glycoproteins, and cell surface membrane. We will also discuss the role of phospholipids and these different components that we have cholesterol, glycolipids, proteins, glycoproteins in cell surface membrane. In the next topic, we'll discuss cell signaling, and we have transport across cell surface membrane that include diffusion, osmosis, facilitated diffusion, active transport, and then we have bulk transport that include endocytosis and exocytosis, and we'll discuss some experiments on these topics as well. So let's see, before we discuss fluid mosaic model, let's see, let's have a look at the behavior of phospholipids. Before we discuss the fluid mosaic model, we should understand the behavior of phospholipids, and we have already discussed the structure of phospholipids in our previous chapter, and that includes let's see, phospholipid consists of three components. There are three components in a phospholipid molecule. It has a glycerol molecule that we can represent with this box. This is a glycerol molecule, and it has a phosphate group attached with it that we can draw here. This is the phosphate group. And we have two fatty acid tails attached with it that we can draw this way. This is one fatty acid chain, and this is another fatty acid chain. Okay, so this is how a phospholipid molecule can be drawn, or we can simplify it showing this part as a head. This is head of a phospholipid molecule, and tails can be represented with this zigzag line. Okay, the head is polar. This is polar head, and these two tails are non-polar tails. Non-polar tails. Non-polar means they don't have any charge on them. There is no charge on these fatty tails, whereas the polar head has charge, and this makes this head water soluble, so this is known as a hydrophilic head. Okay, and hydrophilic means this has an attraction for water, so polar head dissolves in water by making hydrogen bonding. In a polar head makes hydrogen bonding with water. Tails, on the other hand, they are non-polar and they are hydrophobic. Hydrophobic means they are water hitting. So they repel water, or they move away from water. Either they repel water, or these tails move away from water. Now, if we have understood this phospholipid molecule, on the basis of its solubility. The head is a soluble part in water, whereas the tails are insoluble in water. We should understand the behavior of phospholipids in water. If small amount of phospholipids is added into water, if this is a container having water in it, this is water, and we add small amount of phospholipids into it, what will happen? Phospholipids would arrange in such a way that their polar heads will be dissolved in the water. They will be directed towards water, and their tails will be projected out of the water. Okay, so as the tails are hydrophobic, okay, so these are hydrophobic. These tails are hydrophobic. So they move out of the water, whereas these polar heads dissolve in water. Okay, they make hydrogen bonding with water, and this makes a single layer of phospholipids on the surface of water, and this is known as phospholipid monolayer. Okay, if there is a large amount of phospholipids added into the water and it is shaken in the water, for example, if I draw that again, so this container has water in it, and we have large amount of phospholipids added into it, and phospholipids are shaken in this water. Phospholipids would arrange in water in such a way that their polar heads will be outwards, and this will make a globule-like structure. Okay, so polar heads will be outwards, and the non-polar tails will be facing toward the interior of this globular shape. Okay, as is the cross-sectional view. However, if you look at it in a globular way, so this would appear like this would appear as a spherical body with the polar heads on outwards and the non-polar tails to the interior of this globule, and is known as misceles. Okay, now if you look at an animal cell, for example, this is an animal cell, for example. This has cytoplasm on inside, which contain main solvent as water, and outside of this animal cell there is tissue fluid. The main component of the tissue fluid is again water. So there is water on the inside of this cell, and there is water on the outside of cell, and this membrane is made up of phospholipid molecules. So how these phospholipids could arrange themselves in this cell membrane, if there is water on the inside and water on the outside? Obviously the phospholipid molecules cannot arrange themselves in a monolayer. Why? Because if they arrange themselves in such a way that they have the polar heads on one side and the tails on other side, it is not possible. Why? Because there is water on other side as well. So the tails will tend to move away from the water. So as it is water on outside, this is water on here. So the polar heads would obviously make hydrogen bonding with it. So this may make hydrogen bonding and would stabilize this structure, but there is a water inside as well. So they cannot arrange themselves in a single layer. They have phospholipids arranged in a cell membrane in 2 layers like this. Okay, so they're arranged in a 2 layers in such a way that each layer has the polar head facing the water and the tails facing away from each other, and this makes a layer of two layers of phospholipid molecules, which is known as a phospholipid bilayer. This is a phospholipid bilayer. Okay, so in a cell surface membrane, phospholipids are arranged in a bilayer. Then let's see the fluid mosaic model. There has been many different models proposed to describe the structure of a cell membrane, but this fluid mosaic model is most accepted these days. Fluid mosaic model of cell membrane structure. According to fluid mosaic model, fluid means, you know, something that has an ability to flow or that has ability to move. So according to the fluid mosaic model, the phospholipid molecules are arranged in a bilayer in a cell membrane in such a way. So this is a phospholipid bilayer in such a way that polar heads are outwards and the non-polar tails are facing each other and held together by hydrophobic interactions in between these molecules or these non-polar tails. So non-polar tails facing each other due to hydrophobic interactions. Okay, whereas these polar heads are facing outwards in a phospholipid bilayer, as it is, you know, tissue fluid on outside and it is cytoplasm on inside. Both of them, they contain water as a solvent in them. So that's why polar heads are facing towards water and they make hydrogen bonding with the water on outside. So this is water on outside, on outer side of this layer. So this makes hydrogen bonding. You know, these are hydrogen bonding that are present in between the polar head and the water molecules on outside. Okay. According to fluid mosaic model, these individual phospholipid molecules can move within a monolayer. They can diffuse within a monolayer. They are continuously moving, you know. These phospholipid molecules can move from one to the other place. So that is why it is known as fluid. Individual phospholipid molecules can diffuse or move within the monolayer. So that is why this is fluid in nature. That's why it is fluid in its nature. Okay. And then why it is, you know, the second part is mosaic. Why we call it a mosaic model? It is known as mosaic as the proteins are embedded in this phospholipid bilayer. You know, I'll draw that again. And if we look at, you know, the 3D view, This is the phospholipid bilayer from a side view. Okay. The phospholipid bilayer from the side view. And if we look at it from a top view, we can draw it from this side, showing a 3D view. Okay, so this is here, this is the top view. And if we look at it from the top view, you will see there are proteins scattered in it. So these are proteins scattered in a phospholipid bilayer. Maybe they are visible from the top view as shown in this drawing. And you can also see them. They are spanning throughout the entire width of it. And some proteins are spanning only the internal layer. And some proteins are present in this an external layer. And they're scattered here within this phospholipid bilayer. So that is why this is known as mosaic. Okay, and the others from the top view, the polar heads of the phospholipids will be visible this way. This makes an ocean of, like an ocean of phospholipids in which, you know, the proteins are scattered as the glaciers, islands, or you know, the icebergs are scattered in an ocean of water. So that's how this looks like, an ocean of phospholipids in which the proteins are scattered like icebergs, or you know, the islands. So these are the proteins. Okay, so these proteins can be identified as the one that are on the inner side. These are internal proteins, and this is an external protein, and we have this integral protein. These proteins are embedded in the phospholipid bilayer, and this is a phospholipid bilayer. Okay, so this is a fluid mosaic model. In addition to these components, there are some more components embedded in this phospholipid bilayer. The main components include branched carbohydrates attached with these phospholipid molecules or these proteins. Let me copy this structure from here, and this is copied here, and I'll just paste it here again. So let's see. This is the phospholipid bilayer with the proteins embedded into it or scattered into it, making a fluid mosaic model. Some more structures or the components are embedded into it. There are cholesterol molecules that we can see embedded in, you know, this lower monolayer, or maybe embedded in this upper monolayer. Okay, these are cholesterol molecules. We'll discuss how the cholesterol molecules have the same, you know, the head which is polar, and the tail part which is non-polar, and so that is how they are easily embedded in the monolayer. So this is the cholesterol molecule. We'll discuss the role of these components one by one, and then we will see there are branched carbohydrates. This is a branched carbohydrate okay, which is attached with maybe a phospholipid molecule or maybe attached with a protein. And this branched carbohydrate is known as glycocalyx. This is branched carbohydrate. Okay. This may be present on a phospholipid like this. If it is attached with a phospholipid, then this whole molecule is known as glycolipid. And if it is present with this protein here, so this is a protein and a branched carbohydrate is attached with it, then this whole structure is known as glycoprotein. Glycoprotein. So these are the main components of a cell membrane. If I write them, the main component, in largest proportion are the phospholipid molecules, phospholipids. Then the other component is glycolipids and glycoproteins. Then the third is proteins. And the fourth is cholesterol. We'll discuss their role one by one. So let's see the role of phospholipids. The role of phospholipids in a cell membrane. As we discussed earlier, that phospholipids are arranged in a bilayer in a cell membrane. All the cell membranes have the same structure. Either it is cell surface membrane or the membranes of the other cell organelles. They all have the same basic structure. Okay, so these phospholipids are arranged in a bilayer. This is a phospholipid bilayer. Okay, these hydrophobic tails or the non-polar tails, provide hydrophobic barrier. What does this hydrophobic barrier mean? This barrier prevents or inhibits polar charged particles, ions, or water-soluble substances from moving through the phospholipid bilayer. Okay, so like glucose cannot move through it. Sodium ions cannot, move through it. Similarly, the larger other molecules like sucrose, which is water soluble, these cannot move through it. So why don't they move? Due to this, hydrophobic barrier, which is provided by the non-polar tails. However, on the other hand, non-polar substances, gases, and water-insoluble substances, they can easily diffuse through. If there is a high concentration on outside and there is low concentration inside, they can easily diffuse through these spaces from inside out or from outside into, depending upon the concentration gradient. So that is what makes this phospholipid bilayer actually makes this cell surface membrane as a partially permeable membrane. So allows non-polar, okay, or water insoluble and smaller substances to pass through it. So that is why cell membrane is known as a partially permeable membrane. This makes it actually a partially permeable membrane. Partially permeable membrane. Okay. So this is phospholipid. Then the next is glycolipids and glycoproteins. Both they have the same role. We'll discuss in detail in other topics, like they act as, these glycolipids and glycoproteins, even proteins together, they act as receptors for signals or chemical signal molecules, and they act as receptors for neurotransmitters, and they act as receptors for maybe hormones, whereas these glycolipids, glycoproteins, and these proteins may also act as antigen. We will discuss more about antigens and antibodies in immunity chapter. Okay, so antigen. So these antigens actually considered as non-self, recognized as non-self, and they help cell to cell recognition. Okay, so this is the role of these glycolipids and glycoproteins. Now these proteins have the other roles as well. Some proteins may act as enzymes, and the other proteins may be involved in transport proteins. We'll discuss the proteins involved in transport, like channel proteins or carrier proteins, in detail. Then we have cholesterol. Cholesterol regulates the fluidity. Regulates the fluidity of the cell surface, or fluidity of the phospholipid bilayer. Okay, regulates the fluidity of the phospholipid bilayer. So let's see some reinforcement question on these topics that we have discussed, and we will continue further with the structure, the components, and the factors that affect the fluidity of the cell membrane in the next topic. And before that, let's see some reinforcement questions on this topic. Okay, so let's see. This is the first part. Alveolar type 2 cells secrete pulmonary surfactant into the watery fluid that lines the alveolus. The surfactant reduces the surface tension of the fluid so that the alveolus does not collapse. Pulmonary surfactant is a mixture of phospholipids and proteins. The phospholipids form a monolayer on the surface of the fluid. Explain how phospholipid interact with water to form a monolayer on the surface of fluid. For example, if this is an alveolus, okay, so this is an alveolus, and the inner lining of the alveolus is moist. So this is an alveolar epithelium, and this inner lining is moist. So this has a moisture here. Okay, so this is moisture, watery layer, and there are these surfactant arranged on inner surface that make a monolayer. So they would make a monolayer in such a way that their polar heads will be facing towards this surface where the moisture is present, and they would make hydrogen bonding with it, and the tails will be projected. The tails will be projected away from this surface, and this is how this will make a monolayer. This is a layer of surfactant on the inner lining of alveolar epithelium or on the alveoli. So the question is, explain how phospholipids interact with water. As we have discussed, hydrogen bonding of the polar heads with the water molecules, and this makes a monolayer having the tails moved away from this moisture. So we have this marking points. Let's see. Phosphate heads are polar. They are hydrophilic, and fatty tails are carbon chains or tails are non-polar. Hydrophilic polar heads and hydrophobic tails. You can write this. Tails face away from the fluid or water, projecting into the air, except diagram that I have already made. Heads form hydrogen bonds with water. Okay. And max 1 if answer is about a bilayer. This is not about a bilayer. This is about a monolayer. Okay. So this is about a monolayer. So we have made this monolayer. Now let's see, the cell surface membrane of the respiring cells have a role in regulating the exchange of substances between the cells of the body and the blood. Draw a diagram in the space below to show the arrangement of phospholipid molecules in the cell surface membrane. So obviously this would be phospholipid bilayer, and this is for 2 marks. So we have already discussed that the polar head should be outwards. Okay. And the non-polar tail should be facing each other. So this is how we should draw this bilayer. Okay. So let's see, bilayer drawn with heads pointed outwards and tails pointed inwards, and phospholipid drawn with 1 phosphate head and 2 fatty tails. So you see, both of these conditions are fulfilled by this answer. We have this phospholipid molecule with 1 head and the 2 tails, and we have this bilayer. Okay. Now we have another question. Figure 1.1 is a simple diagram of a phospholipid molecule. Explain how the structure of a phospholipid molecule makes it suitable for its function in cell membrane. You may label and annotate figure 1.1 as part of your answer. So you can annotate this is polar head, which is hydrophilic, and this is these are non-polar tails, and these are hydrophobic. So what you have to do is look at this question, explain how the structure of phospholipid molecule makes it suitable for its function in cell membrane. So what are the functions of the cell membrane? It is partially permeable membrane, and it is stabilized in the aqueous medium on outside and on inside. And these are the 2 things that we should mention in our answer. I'm not writing the answer here right now. So let's see if you can understand these points. So you see, max 2 marks for structure, reference to hydrophilic polar phosphate head or group, and hydrophobic non-polar hydrocarbon fatty tails. Reject if labeled correctly but incorrectly described in the text. Okay. So if you have labeled correctly but you don't describe them correctly in the description, you will not get any mark for that, for at least this first mark. A reference to form part of a bilayer. Obviously, you have to talk about bilayer. Fatty acid tails chains may be saturated or unsaturated. We have discussed in the previous chapter the saturated and unsaturated and role of saturated and unsaturated fatty acids in fats and oils, determining the viscosity of the lipids. And then we have head forms hydrogen bonds with water, and this stabilizes the membrane. And tails, idea that unsaturated fatty acid contribute to fluidity. We don't need to exactly to write about directly the fluidity here. We can simply write the hydrophobic barrier, which is provided by the non-polar tail. So that would be just enough, because this is for 3 marks. Then we have the cell surface membrane has a fluid mosaic model. Okay, so describe what is meant by the term fluid mosaic. So this is for 2 marks. As we discussed that fluid is, why it is fluid? Because the phospholipid molecules in a bilayer can diffuse or move around in their monolayer. Why monolayer? Because they don't cross from one layer and they don't move into the other layer. They can keep on moving within the monolayer. Okay. And why it is mosaic? Because the proteins are scattered in the bilayer. So that is why it is, known as mosaic. So that's what describes the fluid mosaic model. In 1934, the biologists Davison and Daniel published their suggestions for the structure of the cell surface membrane as shown in figure 1.1. They suggested that the membrane was a phospholipid bilayer with a layer of hydrophilic proteins on both surfaces. So this is actually a protein layer on outside on both surfaces, on upper surface and lower surface. And we have this phospholipid bilayer which is present here. Okay. The question is, state one way in which the Davison-Daniel structure is similar to the fluid mosaic structure and one way in which differs from the fluid mosaic model. Similarity may be that both have a phospholipid bilayer, both have the phospholipid molecules in them, both have proteins in them. So that may be a similarity. And difference is that in the fluid mosaic model, the proteins are scattered, whereas in this Davison and Daniel model, the proteins cover the upper surface and the lower surface. They make hydrogen bonding with the polar heads. Okay. So similarity is contain phospholipid bilayer. They contain proteins. Details of orientation of phospholipids, lipid bilayer. So that's the same in both of them. And difference is that proteins are only on outside. And in fluid mosaic model, in different locations, discrete different types named or described, or fluid mosaic presence of cholesterol. So this is another point. And this cholesterol has not been shown in this drawing. That's also a part of the fluid mosaic model. And we have the same fluid mosaic model. We have then the list 4 features of cell surface membrane of eukaryotic cell that are not visible in figure 6.1. Where is 6.1? This is the 6.1, which are not which are found in eukaryotic, but that are not visible here. See, number 1 is cholesterol is not visible there. Then the glycolipids and glycoproteins are not visible there. Okay. There are 4 features. Okay. So we have cholesterol not visible, then unsaturated fatty acids that are also found but that are not visible, then carbohydrate chains or glycolipids or glycoproteins are not visible there. These are oligosaccharides, branched carbohydrate, that stand for branched carbohydrate. We can say oligosaccharides or glycocalyx. This is glycocalyx, which is not there. Channel proteins, carrier proteins, peripheral, or we do not need to know these extrinsic proteins or attached to and receptors or antigens. If this is glycolipids and glycoproteins not there, that's just enough. You can write 4 points. No cholesterol. List 4 features of the cell surface membrane of eukaryotic cell that are not visible in figure. So it is cholesterol, it is not visible. Then there are unsaturated fatty acids found in them, which are also not visible in this. And if there had been unsaturated fatty acids somewhere in these fatty tails there should have been kink or this bent shown. Okay, so there is no, they are all around, you know, the straight. So that's why we can write unsaturated fatty acids. Then you can write about glycocalyx, and then you can write about glycolipids and glycoproteins. So that's just glycoproteins. So that's just enough that you can write about 4 features. Explain how the structure of phospholipid allows the formation of a phospholipid bilayer. We have discussed that already. And then that's, you know, drawing of the phospholipid bilayer and the components found in a cell surface membrane. So you can easily identify them. You see, this is N is cholesterol. This is maybe you can say a phospholipid bilayer. If you understand that this hole covers this part, or otherwise you can say this is non-polar or hydrophobic barrier provided by the non-polar tails. This is glycocalyx, and you have this channel protein. Okay, and what else is there? So then this may be a glycoprotein, this whole structure. This may be a glycoprotein, and this may be a, again, glycoprotein. This may be a glycolipid, and these are phospholipid molecules, and this is a cholesterol molecule. So you can identify these different components. Okay, so that's all about it for today. We'll continue with the next part of this chapter. Are you there, Hassan?
Yes, sir.
Great. So that's it for now. See you tomorrow, inshallah.
Allah Hafiz.
Allah Hafiz.